Okay, ladies and gentlemen, I've been informed there is a quorum present, and as it's now 10 a.m., I declare this annual general meeting of Imagion Biosystems Limited open and welcome you to this physical and virtual meeting this morning. My name is Bob Cruz, CEO and Executive Chairman of Imagion Biosystems, and I'd like to thank you for your interest and support of the company. I'm especially delighted, finally, to return to Australia to join my fellow directors and colleagues to host this meeting as a hybrid forum and to make ourselves available for face-to-face. Our AGM is being held on the lands of the Wurundjeri people, and I wish to acknowledge them as traditional owners. I would also like to pay my respects to their elders, both past and present. I would now like to introduce the other members of the board, Mike Harsh, Jovanka Namoska, and Diane Angus, joining me in Melbourne, and David Ludvigsen and Mark Van Asten joining us online. Our CFO and Company Secretary, Jeff Hollis, is also here in Melbourne. I would also like to welcome Robert Miano, a representative of the company's auditor, RSM Australia Partners. Given this is again a hybrid meeting, I just want to run over some housekeeping matters before we commence the formal business. At this meeting, there will be 6 items of business, including 5 resolutions. Each resolution will be proposed and voted on as a simple majority to be carried, except for item 6, which is resolution 5, which is a special resolution. In order for resolution 5 to be passed, at least 75% of the votes validly cast by shareholders on the resolution must be in favor. Today's meeting is being held physically here in Melbourne and online via the Lumi platform. This allows shareholders, proxies, and guests to attend the meeting either physically or virtually. All attendees can watch the live webcast of the meeting. In addition, shareholders and proxies have the ability to ask questions and submit votes. Online attendees can submit questions at any time. To ask a written question, select the messaging tab at the top of the Lumi platform. Type your question in the box towards the top of the page and press this arrow symbol to send. A copy of your submitted questions along with any written responses from our meeting team can be viewed by selecting My Messages. To ask your question verbally, pause the broadcast on the Lumi platform and then click on the link under Asking Audio Questions. A new page will open where you can be prompted to enter your details before being connected. You will listen to the meeting on this page while awaiting to ask your question. Please note that while you can submit questions starting now, I will not address them until the relevant time in the meeting, when I will also address any questions from those physically in attendance. Please also note that your questions may be moderated, or if we receive multiple questions on one topic, they may be amalgamated together. And finally, due to time constraints, we may run out of time to answer all of your questions. If this happens, we will answer them in the due course of time. Voting today will be conducted by way of a poll on all items of business. In order to provide you with enough time to vote, I will shortly open voting for all resolutions. At that time, if you are eligible to vote at this meeting, a new voting tab will appear. Selecting this tab will bring up a list of resolutions and present you with voting options. To cast your vote, simply select one of the options. There is no need to hit a submit or enter button, as the vote is automatically recorded. You do, however, have the ability to change your vote up until the time I declare voting is closed. I now declare voting open on all items of business. The voting tab will soon appear. Please submit your votes at any time. I will give you a warning before I move to close voting. Before attending to the formal business of the meeting, I would like to take this opportunity to reflect on our accomplishments of 2021 and provide some insight regarding what we anticipate for the near future. Our MagSense technology is designed to provide non-invasive detection of cancer and other diseases without the risks associated with radiation-based imaging technologies. Additionally, by using cancer-specific targeting molecules, we aim to eliminate unnecessary biopsies that are performed when standard imaging identifies a suspicious lesion, but can't confirm if it's malignant or benign. The prospective value and benefit to patient care of our approach was recognized by the FDA with the breakthrough device designation we received in 2019. And bringing this first-of-its-kind innovation from concept to clinic has been our sole mission. With that mission at the forefront, in 2021, the majority of our efforts were directed towards supporting the phase 1 study of our MagSense HER2 breast cancer imaging agent being taken here in Australia. While the study had officially commenced in December of 2020 at Monash Hospital in Melbourne, we found it difficult to secure additional clinical sites the first half of the year as hospitals dealt with the pandemic. Additionally, we did not anticipate the substantive and long-lasting impact of the pandemic would have on clinical research at many of the hospitals and the reduction of patients visiting their doctors. However, in the second half of the year, we were able to add new sites in New South Wales and Queensland, and we had multiple patients enroll in the study. Admittedly, enrollment continues to be challenging. As things return to normal, we anticipate that we will see an uptick in patients being screened and therefore accessible to our study and investigators. Over the course of 2021, we received many inquiries and calls for more frequent updates on the study, so I'd like to take this opportunity to clarify our view, consistent with ASX guidelines, regarding responsible reporting to shareholders. We understand completely that shareholders are anxious to know how the study is progressing, and we feel the best way to do that is when we have a meaningful information to communicate, such as our March announcement of interim results. We will continue to provide progressive updates when we have materially relevant information to share. Last year at this meeting, I mentioned that getting the first human data from our phase 1 study would be an important inflection point for the company. So we were very pleased to achieve that objective and report in March the interim results from the first 5 patients. Even though we had every expectation our imaging agent would be safe and well tolerated, it was great to see that borne out in the assessment from the first 5 patients. Many new drugs fail in phase 1, and even though our imaging agent is not a drug per se, it is an injectable and is therefore subject to the same concerns as any substance injected into humans. So indeed, the safety and tolerability observations from the first 5 patients is a positive. Additionally, while results from 5 patients are not enough to determine the effectiveness of the nanoparticles for detecting metastatic disease, the data from these first patients has served to provide evidence that the nanoparticles are functioning as expected and has encouraged us to continue the study through this year to ensure we have obtained sufficient data. From the interim assessment showing that the nanoparticles can reach the target lymph nodes, we believe we are one step closer to demonstrating that targeted magnetic nanoparticles can be used to non-invasively find and detect the presence of cancer. Having demonstrated that we could design and develop a targeted nanoparticle for breast cancer, and having achieved entering the clinic, in 2021, we also began to expand our R&D efforts to look at other forms of cancer, where our targeted nanoparticle technology could also add clinical value. We received a $50,000 grant from the Australian government to fund research with Monash University's Biomedicine Discovery Institute to kickstart preclinical research on prostate cancer as an imaging agent. This work is ongoing, and as we noted in our most recent quarterly activity report, we have made progress with the PSMA targeted nanoparticle. This imaging agent, like most radiopharmaceutical PET tracers, utilizes a molecule to target the prostate-specific membrane antigen, which is expressed in most forms of prostate cancer. But unlike PET tracers, our MagSense technology uses our biosafe magnetic nanoparticles instead of radioactive tracers. This we expect will making it safer as an alternative for prostate imaging. As we continue our preclinical research this year, we hope to enter 2023 with a clearer picture of the feasibility and timeline for developing a prostate cancer imaging agent for clinical use. Also in 2021, we formed a collaboration with Patras Limited, an ASX-listed biopharmaceutical company, to explore the possibility of developing an imaging agent for brain cancer. The collaboration pairs the targeting capabilities of Patras's deoxymabs with the imaging capabilities of the MagSense technology. As a result of the progress of our in-house research in 2021, we are expecting this year to expand the collaboration to include a prominent Australian educational institution that has expertise in animal models of a severe and prevalent form of brain cancer, known as glioblastoma, to further this research. In addition to our R&D pipeline focused on improving diagnostic imaging of cancers, we have also cultivated third-party relationships where our expertise in nanoparticle technology can be leveraged to realize future revenues. We do not expect to see material changes in revenue for this year, but we'll continue to work with our partners as they develop their biomedical products, building an underlying source of revenue based on our nanoparticle manufacturing capabilities. We ended 2021 in good financial shape with a cash balance similar to the start of the year, in part due to the strong support from our shareholders exercising their listed options and the receipt of our 2020 R&D tax credit. Over the last 2 years, we've made significant progress in transitioning to be a clinical stage company, something many biotech companies fail to achieve. We have shown that we can make a targeted magnetic nanoparticle imaging agent. We have dosed breast cancer patients with the imaging agent, and to date have shown it to be safe and well tolerated. As we continue to accumulate the evidence of its effectiveness as an aid in the clinical care of breast cancer patients, we expect to further expand our pipeline to exit 2022 with a robust plan for developing molecularly targeted diagnostic imaging agents that use our magnetic particles to provide earlier detection of cancers and other diseases. In closing, I'd like to thank all the Imagion stakeholders, shareholders, customers, collaborators, advisors, employees, and directors for their support, and remind all of us why your support is so important. While treatments for cancer have improved over the past few decades, and the pharmaceutical industry puts massive resources behind finding treatments, we still don't have the proper tools to detect cancer early enough to enable treatments to work. The markets are large, and the opportunity to play a leading role in improving the way disease and cancer are diagnosed and treated is directly in front of us. Although our first study has progressed more slowly than we would like, and it can be tempting to see the glass as half full, we view the interim results positively and believe we are on the right course and will be able to positively impact patient outcomes in the future. I will now move on to the formal business of the meeting. Item 1, financial and related reports. This item requires us to receive and consider the financial report of the company and its controlled entities and the related directors and auditors' reports in respect of the financial year ended 31 December 2021. There is no requirement for shareholders to vote on these reports. Are there any questions or comments on the report from the room or online?
Nothing.
The next 5 items of business require a shareholder vote, and will be conducted by an online poll as previously mentioned. Item 2 is Resolution 1, the approval of the remuneration report. Item 2 is considered, and if thought fit, to pass the following resolution as a non-binding ordinary resolution. That the remuneration report of the company and its controlled entities for the year ended 31 December 2021 be adopted. Please note that a vote on this resolution is advisory only and does not bind the directors or the company. The board abstains from making a recommendation in relation to this resolution. There is a voting exclusion on this resolution. The key management personnel, as set out in the company's 2021 annual report, and their closely related parties must not vote on this resolution. An exception to this prohibition exists where the person is exercising a directed proxy, which specifies how the proxy is to vote on the proposed resolution on behalf of someone other than the key management personnel or the closely related party. A key management personnel or closely related party that is appointed as a proxy must not exercise the proxy on a resolution connected directly or indirectly with the remuneration of a key management personnel if the proxy is undirected, that is, if the appointment does not specify the way the proxy is to vote on the resolution. However, the prohibition will not apply if the person is the chairman of the meeting at which the resolution is voted on, and the shareholder expressly authorizes the chairman to exercise the proxy even if the resolution is connected directly or indirectly with the remuneration of the key management personnel of the company. I report that the valid proxies received in advance of the meeting are displayed on the screen. Are there any questions or comments in relation to item 2 from the room? Mr. Hollis, is there any further questions or comments submitted online?
No questions.
I now remind you to register your online vote either for or against in relation to item 2 concerning the approval of the company's remuneration report. Item 3, Resolution 2, the re-election of Michael Harsh as the director. Item 3 is to consider and, if thought fit, to pass the following resolution as an ordinary resolution. That Mr. Michael Harsh, who retires in accordance with Clause 20.3 of the company's constitution and ASX Listing Rule 14.5, and being eligible, be re-elected as a director of the company. The directors of the company, other than Mr. Harsh, recommend that the shareholders vote in favor of this resolution. I report that the valid proxies received in advance of the meeting are displayed on the screen. Are there any questions or comments in relation to item 3 from the room? Mr. Hollis, any further questions online?
No questions, Bob.
I remind you to register your online vote either for or against in relation to item 3 concerning the re-election of Mr. Harsh as a director. Item 4 is resolution 3, the re-election of Jovanka Namoska as the director. Item 4 is to consider and if thought fit, to pass the following resolution as an ordinary resolution. That Ms. Jovanka Namoska, who retires in accordance with Clause 20.3 of the company's constitution and ASX Listing Rule 14.5, and being eligible, be re-elected as a director of the company. The directors of the company, other than Ms. Namoska, recommend that the shareholders vote in favor of this resolution. I report that the valid proxies received in advance of the meeting are displayed on the screen and ask if there are any questions from the room. Mr. Hollis, any questions from online?
No questions, Bob.
I now remind you to register your online vote either for or against in relation to item 4 concerning the re-election of Ms. Namoska as director. Item 5, resolution 4, EIP capacity. Item 5 is considered and, if thought fit, to pass the following resolution as an ordinary resolution. That the employee incentive plan rules, as approved by the 2020 annual general meeting, be amended to change clause 6A6, to read as an offer made under a disclosure document as defined in the Corporations Act would exceed 10% of the number of shares on issue at the offer. There is a voting exclusion on this resolution. Any directors of the company and associates of directors must not vote on this resolution. The directors of the company recommend that shareholders vote in favor of this resolution, and I report that the valid proxies received in advance of the meeting are displayed on the screen, and I ask if there are any questions from the room on item 5. No questions from the room. Mr. Hollis, any questions online?
No questions, Bob.
Thank you. I now remind you to register your online vote either for or against in relation to item 5 concerning the EIP capacity. Item 6, resolution 5, approval of the 10% placement facility. Item 6 is to consider and, if thought fit, to pass the following resolution as an ordinary resolution. For the purposes of ASX Listing Rule 7.1A, and for all other purposes, approval is given for the company to issue up to an additional 10% of its issued equity securities by way of placements over a 12-month period, on such terms and conditions more particularly described in the explanatory memorandum and accompanying the notice of AGM. Item 6 is a special resolution. Accordingly, at least 75% of votes cast by shareholders present and eligible to vote, in person or by proxy, at the meeting must be in favor of this resolution for it to be passed. There is a voting exclusion on this resolution. Any persons who are expected to participate in or who will obtain a material benefit as a result of a proposed issue of shares under the 10% placement facility accept a benefit solely in the capacity of a holder of shares, and associates of those persons must not vote on this resolution. The directors of the company recommend that shareholders vote in favor of this resolution, and I report that the valid proxies received in advance of the meeting are displayed on the screen. Are there any questions or comments in relation to item 6 from the room? No questions. Mr. Hollis, any questions online?
So there's no questions in relation to item 6, but there are other questions which might be a good time to address them now.
Okay.
Before we get to the final voting.
Okay. Firstly, I remind everyone to register your online vote either for or against in relation to item 6 concerning the approval of the 10% placement capacity. So before we move to close the voting, let's go ahead and open the questions from online.
So first one from Anesti Lazaridis. Has the company made a final decision on the cohort number required for the current trial, i.e. 10 or 15 participants?
So the answer to that is no, we haven't made a final determination. We designed the study to be open up to 30 patients to give ourselves the flexibility to continue to investigate as we saw fit. At this point in time, we believe that achieving somewhere between 10 and 15 patients will give us sufficient information to make a decision on it for going forward. But we don't yet know specifically whether 10 will be enough or 15 will be enough.
Thank you. Another question from Mr. Abhishek Prasad. What is the realistic timeline that we're looking at completing the trials? Also, does loosening of restrictions help with the trials?
So as I mentioned in the chair remarks, we are hopeful that the loosening of restrictions as we're coming out of the pandemic, so to speak, will in fact improve the ability for patients to access their caregivers, be screened for cancer, and potentially be available for the study. It's still very hard for us to predict the rate of recruitment at this stage. We have 4 sites that are all actively screening patients. We meet with them on a weekly basis to try to look at what their screening numbers look like. We are still hopeful that we can achieve meaningful numbers of study subjects during the course of 2022, but it's not something that we're really able to predict at this time.
Thank you. Further question, of the patients that have completed the trial, why can't the investigators or the board tell shareholders whether they can detect cancer?
Yeah, so this gets to that question associated with how many patients do you think you need to do this? We, at the interim results that we reported for the first 5 patients, what we are able to report is our observations from that, but we're not able to draw conclusions from an N of 5. And so what we're really, it would be imprudent of us to try to suggest whether we can determine effectiveness at 5 patients or at 6 patients or when we get 1 more patient. So what we really are trying to achieve is that we have enough evidence that it's pointing us in a direction with confidence. And today what we've been able to say is that we're very encouraged by what we see with the first 5 patients, but we can't make an individual call on any given 1 patient. And so it's not appropriate for us to report what we see on 1 patient versus another. We have to wait until we accumulate enough information to be able to say, is this looking like it's moving in the right direction that we can ultimately submit to the regulatory authorities to get clearance for its use as a clinical aid.
Yep, thank you. And another couple in relation to the trial. In light of the protracted phase 1 clinical trial, is the board and or management confident in our ability to expedite the anticipated follow-on pivotal trial?
So again, referring back to the remarks that I made, while we can't draw conclusions as to whether or not, answering that prior question, we can say we can detect the cancer or not. What we can say is that we're significantly encouraged by what we see in the study so far. And as a result of that, in fact, we are starting to make plans for the next phase of clinical involvement. We're encouraged enough that we believe that it's time to start to progress towards what the next phase would be, moving towards a pivotal study. And so I think that the short answer to that question, Mr. Hollis, is yes, in fact, we believe that we have enough evidence from what we're doing to A, continue the study here in Australia, and B, begin to make plans for the next phase of testing.
Yeah, I think this flows in. Mr. Robert Scanlon's asked a question online. What are the immediate next steps following a successful trial?
So immediate next steps would be to go beyond Australia, to move into jurisdictions where we can then look for US and or European regulatory authorization as well. So what we'll be doing then is making additional HER2 imaging agent to be used in larger studies that would be done both in Australia as well as elsewhere.
And another question from Mr. Scanlon: what are we still needing to learn from the current trials?
So probably, I think the shortest answer there is while we have evidence that the nanoparticles are reaching the lymph nodes, what we need to know is what's the dose, for example. That's one question we still need to answer. Can we reduce the dose of nanoparticles, the amount of nanoparticle material we give, and still get the same result? Do we need to potentially increase it? Do we need to wait longer periods of time before we do the imaging? So today in our study, we image at 24 hours and 72 hours post the injection. So it's at this stage, some of the things that we still need to learn are how best to optimize. What we know now is that we can inject the imaging agent. It seems to be safe and well tolerated. It is getting into the lymphatic system, but now we need to look at how best to optimize it. So when we go do multiple hundreds of patients to get clinical approval, we're actually doing it with the most optimized administration and protocol.
Thank you. Moving away from the trial, recent company announcements have had a high degree of ambiguity, and I'm presuming this is coming from our March trial update, especially, where more questions are raised than answered, and shareholders are left looking for hidden messages and attempting to read between the lines.
So I guess I would respond to that with, don't look for hidden messages, and we're not trying to be obtuse. We're very careful about reporting the facts, right? And we've said repeatedly, and what we've said in my chair remarks is that we're going to continue to disclose things that we consider to be material. And so I wouldn't try to read between the lines. If we report that we feel that in this example, that the evidence is suggesting that through the first 5 patients, we're seeing that it's safe and well tolerated, there's nothing more than that to it. And if we say that we are not yet ready to make a determination whether it's effective, it's because with an N of 5, we're not capable of making that conclusion. So I would encourage shareholders not to think we're trying to be coy, we're not trying to be cutesy. We're simply reporting what we can report that we see as being material information for the company.
Thanks, Bob. Another question from a shareholder. A statement and then a question. Why has the news flow dried up since November 2020? There have been few price-sensitive announcements in the last 18 months. Shareholders are of the belief that there was more going on in the background, which would lead to further news flow. Aside from the new facility, is there anything newsworthy on the horizon?
So I would say 2 parts to that. One is that, again, we are very clear that we make announcements when we think there's something material. So if the news flow feels like it's dried up, according to the question, it's because perhaps nothing material has happened. It doesn't mean nothing is going on. It doesn't mean that we're not undertaking R&D activities or that we haven't made progress, but it may not be material. And we're very careful not to just put out news just for the sake of putting out news. I would caution folks to look at no news as not being bad news. It just means that we haven't had anything material to report. We do include, as part of our communications, investor newsletter, which does provide insight and information as to activities that we have that may not be material by themselves, but we restrict our ASX announcements to those things according to ASX listing rules are material to the company. As for forthcoming, it's hard to predict when something material might actually happen, but we would say certainly that our progress with the development of the current clinical study, any meaningful progress for any of the other preclinical activity that we have, and or activity related to expanding the clinical study in jurisdictions outside Australia, we'll report them as they take place.
Thanks, Bob. Another question here. This might be more relevant to you, Di. I'll read a bit of the background. An independent non-executive chair is fundamental to good corporate governance, improving performance and accountability of the management team and reducing the potential for critical conflicts of interest. It is also a required feature for many institutions to invest in a company. Does the board of directors plan to appoint an independent chair in the future?
Thanks, Chair. It's an important question. The company, through the Rem Nom Committee, actually conducts a separate chair performance review. So we go through an annual review of how the chair is executing that role as an independent chair as such, and then separately as a CEO. So we conduct 2 performance reviews. We are very mindful of our particular set of circumstances. But having said all of that, it is something that the company is looking to, reviewing, thinking of, and planning down the track to move towards that model.
I think, Bob, it's fair to say it's not uncommon in the US.
It's not uncommon in the US, but again, we're an ASX listed company with a substantial shareholder base in Australia. And so if we don't look like the normal zebra that they're used to seeing, I think it's a fair question to ask. And as Di, as chair of the Rem Nom Committee, has responded there, it's something that we do take a look at ongoing, and I'll be the first one to recognize if I don't think that it suits the company appropriately.
Yeah. Another question here. This is probably a better place for me to answer, actually. Why haven't any directors bought shares on market recently or generally historically? It's a really good question, that, and I think some shareholders are of the view that they like to see directors buying on market. Unfortunately, when directors buy on market, they can sell on market too, and that's not viewed as well. And other shareholders like to see directors actually not buying as many shares and being purely independent. So we really leave, there's no recommendation from management to any of our directors to be going and buying shares on market. That is purely their prerogative and for their decision. I guess another part to that question, do they see the shares as good value? We don't comment on the value of the shares and the share price, but what I can say is, just an outsider, having been involved in many listed companies, as a small company in the position we're in, we have a very high caliber board, and they wouldn't be giving us their time and effort and energy and commitment and putting their name to us if they didn't believe in the story. And I think that's more important than them actually going and buying 10,000 shares on market tomorrow to be seen to support the company in that way. They're doing far more than that, just being with us. So that's an answer to that question. The last question we have, and again, this is probably better for me to answer as well. Please explain the process of how the board, and this is from Mr. Ruben Pack. Thank you, Ruben. Please explain the process of how the board determines it is in compliance with its continuous disclosure obligations. Look, really good question. We have a board with some directors in Australia and directors in the US. We're an ASX listed company. Look, we meet regularly as a board. We have our general counsel, Harry Kingsley, who's in the room today from K&L Gates. He attends all our meetings and is available on call. I myself have listed company experience spanning 14 or 15 years now, and keep up to date with all the continuous disclosure obligations of the ASX and the Corporations Act. We have Di and Ivanka both on listed company boards in Australia as well, to ensure we've got that good representation at a board level. Bob himself did the Australian Institute of Company Directors course not that long ago. And Mike and David and being, even though they're US based, have a lot of listed company experience in the US, doesn't translate directly to Australian laws, but is not that dissimilar in a general what's material, what's not material, how should we be disclosing information. And in addition to that, we take very seriously our disclosure obligations, and therefore have a disclosure committee that meets regularly whenever we need to in terms of discussing any potential announcements. So anything further from anyone else on that one? Or, I conclude that is the last of our questions, unless there's any more in the room.
Yeah, thanks. Just taking you, John Louis, by the way, just taking you back to the discussion about the trial that's currently being conducted. My observation is that the trial is actually quite detailed in terms of not only trying to determine the safety and efficacy, the safety of the nanoparticles, but also potentially provides some read on its ability to do what it's supposed to do. And so I do understand that currently you don't have sufficient statistical power to be able to say a lot about it. My question is whether whether you expect to have discussion of its efficacy. That's any information, pardon me, that's yielded from the trial when you get sufficient number of enrolments.
Yes. Short answer is yes, absolutely. So just taking a step back on that question, right? We could have done a typical phase 1 study of safety only, right? Safety and tolerability, taking non-breast cancer patients, naive patients, just see if we inject this imaging agent, does it hurt anybody, et cetera, right? But we wouldn't have learned whether or not we have the prospect of an imaging tool for detecting lymph nodal involvement. So that's what made the initial study a little bit more complicated than typical phase 1 studies. We really we had every expectation that it was likely to be safe and well tolerated. So just doing a phase 1 safety study wasn't going to move the needle for us very far. So the study, in fact, was designed to try to accomplish some measure of effectiveness and understanding while still demonstrating for ourselves that, in fact, our expectations of safety and tolerability were well founded. So that's exactly what we're trying to get to, is can we get to, and whether that N is 10 or 15, whatever we need in order to be comfortable that, in fact, what we're demonstrating is that we've designed a targeted magnetic particle that, if injected into a woman's breast tissue and picked up by the lymphatic system, is able to determine whether or not there's tumor involved in a lymph node. And so we expect that with somewhere between 10 and 15 patients, we're likely to be able to get that answer. And then we may still need to do, as I said earlier, dose optimization to make sure that we get, for the future product, that we really get that right. But what we would say today is that we have evidence that suggests it's moving in the right direction. If we felt that it wasn't working at all, we would stop the study. Our investigators would ask us to stop the study. There's no point in dosing patients if this got no chance of working, right? Can I say today that, we'll say today with the N of 5 that we've reported interim results on, that everything is exactly the same? No, there is some variation across patients in what we are able to see, and that's expected. So that's why we need a bigger number. But the outcome of the study, when we achieve enough numbers of subjects, is in fact to allow us to make the decision, should we move this forward as an imaging agent because we got evidence that it's going to work. Anything else, Di? You're very experienced in clinical studies, so.
All it is it's situational law for clinical research. Very typically, you have to wait to the complete conclusion of a trial before you can speak, say anything, because the risk of misleading in a negative way as much as a positive way. So we're very respectful of our shareholders that we're so anxious for these results as well, but are very mindful that early interpretations are fraught with danger. So we take an optimistic yet, as proactive approach as we can. And as Bob has said, when you start a trial, you start off with a number in mind, and you have to have a kind of a priori approach and then stand back from it as a very independent and well-run trial design. So as soon as we can, we will.
Yeah.
And we're looking forward to it very much.
And each new patient adds that little bit more information, but each new patient by itself is just another N of 1. And so we're very cautious. I mean, we get a lot of the investors that often ask, well, how many patients have you had now? When's the next patient, kind of thing? And it's on an individual basis, it's kind of like you look at a tree, but what we're waiting to see is the forest. And so hopefully, we'll get to that point where we can actually then take a look at the forest and say, what does this really look like? It's not helpful to report on an individual tree, so to speak.
A follow on. In hindsight, would it have been better to run a simple, regular phase 1 trial? Is it safe?
My answer to that is no. We could have potentially gotten through a larger number of patients that says, yep, safety and tolerability is good, but I still don't know. If you take a naive patient, for example, and you inject our imaging agent into them, and it goes into the lymph nodes, it'll tell us safety, tolerability, etc., but it won't tell us a thing about whether or not we could detect lymph nodal involvement. And that's the question we really wanted to de-risk, right? Because I don't want to go try to design a study for 200 or 300 patients if I don't have really good confidence that it's going to get us what we want. And just testing naive patients for safety would never give us an answer whether or not I have a low risk of being able to detect tumor involvement.
Bob, I think the other thing is the fact that we had a limited number to start with allowed us to really understand the kinetics of this and understand how long should we wait between further imaging exams, so we can go back and modify the protocol, be much more efficient going forward.
And also, if I can add,
Yeah,
we love this question.
Yeah.
But I think it's, the challenge and the beauty of the trial design that our nanoparticles are decorated with specific antibodies to a specific target. And that target's only presented on these type of cancer cells. So going halfway and saying we think we can see the pharmacokinetics and dynamics of how the nanoparticle passes through the body is good, as Mike has said, but the real prize here is that not only can you get through the body in a reasonable way and in a reasonable quantity, but actually attach to, prospective or suspected cancer cells in those lymph nodes. So that's what this is really about. So would it have been simpler? I think as a practical matter, probably not, because the patient has to go to the injection, has to be in the clinic, has to have MRI. There's still these procedures. And But I take your point because we are dealing with very, brave and terrifically admirable women who are undertaking this with a diagnosis of breast cancer. So they're all heroes in our eyes.
Yeah.
And but I understand the question.
Thank you.
Anything else? Yes, sir.
Yeah, great. Well, it seems that we're short on patients here in Australia for phase one. Why can't we go to America?
So this gets to why so many companies do in fact come to Australia for phase one type studies, and that is that the regulatory and clinical ethical environment here is very conducive to phase one studies. But the stringency in, for example, the United States, doing it with the FDA or in the European Union, makes it much harder. So the material that we made, the imaging agent that we made, and the rules associated with getting the study done here made it easier for us to get the study done in a more timely manner and less expensively. If we were to turn around and try to do this in the United States, we would have to make a new lot of material that met FDA requirements, and then we'd still have to go through the regulatory approval process in the United States. So here you have an ethics committee that approves a study, and it's good for any institution that's within the purview of that ethics committee. In the United States, you have first to get the FDA's clearance, and then you got to get every institution to sign on with their own ethics review and their own what's known as an institutional review board. It's possible to do, but it's more time consuming and more expensive. And we've just felt that we could still achieve what we needed to achieve here in Australia, but we certainly have been impacted by the number of people being able to get into the hospital.
That would be the same as European.
It would be, yeah. European regulations are very similar to the United States, very stringent. Yeah, we have looked elsewhere. Admittedly, our team looked at a variety of different countries, and the problem is every country, bit of an exaggeration, most countries are different. So how you would do something in the United States, you'd have to do something different in China. You'd have to do something different in India. So we really did try to scour and see if what we've done here in Australia could be moved to another jurisdiction, but all the rules are different, and so it sort of meant starting all over again. And we just felt that we would spend a lot of time and a lot of money trying to get started elsewhere. If we just sat pat and stayed here and persevered in Australia, we'll still get to what we need to get to.
Yeah.
Okay. Any further questions? Come in.
Nothing else in the room? Yeah, nothing else online.
Thank you for your questions. This brings us to the end of our formal business. I now ask you to complete your voting paper and to place it in the ballot box here in the room. For those joining virtually, please note that the online voting will remain open for a further 2 minutes, and I will then formally close the meeting. The results of the poll will be notified to the ASX and put on the company's website later today. I'll now ask Mr. Hollis if there's any further questions coming in online.
There is a couple of other questions coming in online. This one's worth answering. I've been asked this a lot in the past. Any further revisiting to incentivize patients for treatment, for their participation, more than just covering costs?
No. We've covered their costs. So if we need to put them up in a hotel or pay for transportation, we have included compensation for their time according to the normal ethics capabilities. But it's really frowned on both by the institutions, so all the hospitals that we work with and the clinical investigators, and generally by the Australian Biotech Association and by ASX, to try to incentivize what are known as vulnerable populations. It's really frowned on to try to incentivize vulnerable populations. And it's quite clear that someone who has just been told you have breast cancer would be considered someone vulnerable. And so to incentivize them to participate in the study is discouraged by the normal ethics review and certainly discouraged by our investigators. Their primary focus is on the care of that patient, and they don't want to try to compromise the care of the patient by having them have a financial incentive just to volunteer.
Yeah. One further question as well. This from Robert Scanlon again. and I think I share his. I'm still not very clear exactly how long the trial is expected to last, i.e. to get to completion. Do we have a run rate to give some more accurate timelines? I think, unfortunately, we would love that.
Yeah, we'd love to be able to do that. What happens is sometimes it comes in fits and starts. You may have 2 or 3 patients present to one hospital in the course of a week, of which one might be eligible, and choose not to participate. So the problem is that we don't have control over the screening of breast cancer. We don't have control over who presents at the hospital and what their willingness and individual circumstances are. So it's really difficult to participate. What I can say is that based on our conversations with our clinical sites, the numbers of patients presenting to the hospital is starting to improve compared to what it was 6 or 9 months ago. And so we are looking at that as potentially being encouraging for potentially seeing screening and possibility of recruiting, but it's really impossible to predict. As Di said earlier, we're very grateful to those who do participate because they are really selflessly volunteering. This is someone who has just been diagnosed with breast cancer, is expecting to start treatment in the course of typically between 5 and 10 days post-diagnosis, and has then volunteered to participate in our study in that interim period of time. Many people just say, I just want to get on with treatment. I don't want to think about anything else. So it's not something we can predict in terms of who is willing to volunteer. What we do know is that our clinical investigators are very practiced at having this conversation with their patients. And so we really are hopeful that they can continue to recruit into the study for us.
Beautiful. No further questions.
Okay, with that, I think our 2 minutes has passed, and so I can confirm voting is now closed. And I'd like to thank everyone, our shareholders and directors and all, for your support, and fully close the meeting. Thank you.